All right, welcome everyone. Thanks for joining us for this Coastal Arctic Biogeochemical Data Synthesis Workshop for Arctic Colors. On behalf of the organizing committee, I thank you for coming. And so we know that there are a variety of processes that are changing in the Arctic very fast. We discussed some of this yesterday. Many of you are aware of this, and so I will focus my introduction mostly on Arctic colors at this point for the sake of time. So Arctic colors as a project aims to quantify the coupled biogeochemical ecological response of the Arctic nearshore system, and that's a very important word, to rapidly changing terrestrial fluxes on one side, ice conditions on the other side and across, in the context of both short-term and long-term change. And so the Arctic Colors overarching science aims are to establish the effects of land, ice, and future change, both as warming land and melting ice on nearshore Arctic biogeochemistry. And here on the right-hand side, if you can see my cursor, I'm looking on the bottom panel. In light pink is the domain or study region envisioned for Arctic colors. And from the river mouths of both large rivers such as the Yukon and Mackenzie to regionally important smaller rivers, which is on the top panel at a greater magnification, onto this nearshore region, including smaller tundra rivers, coastal lagoons, and erosional bluffs. So Arctic Colors is a proposed NASA field campaign to, as I've already mentioned, improve understanding and prediction of the rapidly changing coastal Arctic. Since proposed in 2014, Arctic Colors has developed a science plan, which you can see here on the bottom left, an international team of collaborators, which you can see in this spreadsheet here, as well as a project website, which you will see repeatedly mentioned in my presentation, so that you can go there and get more details. A fair amount of outreach materials. And we've had about more than 20 meetings so far of different types, sessions, workshops, etc., to reach out to you over the course of since we got started. So the science plan includes a variety of measurements and variables to be measured, which is the point of this cartoon. From water column to surface to bottom to air to land, from satellites, ship-based, land-based, autonomous, a variety as well as models, aspects for which we are addressing in this workshop. It's a highly coordinated and integrated effort that is being proposed. For this workshop, though, and this current project on the Arctic Colors data synthesis facilitated by OCB, I forgot to say this at the very beginning. This data synthesis project is coordinated and funded by the OCB office, as well as by the NASA OBB Ocean Biology and Biogeochemistry program. Very important. The aims of this data synthesis effort that you are now participating in is to provide a comprehensive synthesis of existing data in the northern high latitudes. We have to focus. We'd like to do everything, but it is primarily on carbon stocks and fluxes and their optical proxies. There will be other biogeochemical variables and processes to be included in this data set, whatever possible. The plan is to collect and combine existing in situ data, modeling results, so in satellite data sets. Hence the 3 days of this workshop, yesterday focusing on in situ data, today on models, tomorrow on remote sensing. To, as much as possible, resolve certain differences in reported data due to methodology and create and host a newly synthesized database specific for Arctic colors. There are many others, yeah. And with this, identify data and knowledge gaps. These aims go beyond the 3 days of this workshop. Becoming more specific, for this meeting, the goals are to identify these field data sets, the modeling activities that happen today, and remote sensing capabilities tomorrow within the Arctic color study domain. And among all of us to contribute and share data, especially those which are upcoming, not only those which are already archived. And if at all possible, to let the organizers know about these data sets before November 30th of this year, so that we can continue the synthesis. The meeting agenda. Day 1 was focused on field measurements, as I already said. Today, we will focus on modeling activities with presentations highlighting major modeling efforts in the coastal Arctic. Tomorrow, the focus will be on remote sensing. Let me tell you a little bit of what was presented and covered yesterday, day 1, for those of you who perhaps did not attend. There were 4 presentations. They're already online, so I encourage you to go watch them as I'm giving you but the briefest details. Oh, I can see that 2 here joined. So the first presentation was an introduction to Arctic colors in the OCB data synthesis activity. Okay. The second presentation, which my number 2 disappeared, was on the Arctic Great Rivers Observatory. And in there, some of the conclusions reached were need for standard protocols, access to all data, that small rivers matter, seasonality matters, scaling matter. And there was a fair amount of discussion of the issue that flux to estuary is different from flux to ocean, addressing this continuum. The third presentation was an update on the Beaufort Lagoon ecosystems LTER. And in there, some of the highlights were issues with long-term versus short-term sampling. The synergy that exists with Arctic colors with respect to, for example, small rivers, erosion, and other things, and the importance of shallow and deep groundwater discharges, as well as the timing of releases. The fourth presentation was on the Malina and the Nunatario projects. In the case of Malina, many publications and all the data are available. And then highlights included the relevance of autotrophy versus heterotrophy, photochemistry, of course, and the issue of colorless DOM. And then we finished the day with 6 breakout rooms. There were many highlights. I'm here showing just a few. The fact that there are many data sets available and these were identified in the various groups. Nonetheless, estuaries are data deserts, not deserts. There was little discussion on snow and ice data, but that perhaps reflected the composition of the attendees more than anything. Repeatedly, there was mentioned the need to optimize an Arctic color data repository, as well as the need for Arctic-based data for model parameterization and validation, of which I am certain we will hear more today. The need for establishing and adopting calibrations and protocols, and etc. So without further ado, the four speakers, let me introduce the four speakers today. Gabby Negrete-Garcia from Scripps will talk about the community model. Joel Rowland will follow talking about interface. Mike Rawlins will talk about DOC exports from the Alaska North Slope to the Beaufort Sea. And last but not least, Blake Clark will talk about the FVCOM-BGC-optical model, focusing on the Yukon coastal Arctic. Each one is going to be 15 minutes. I want to tell the speakers, today's speakers, that yesterday's speakers were awesome. They were on time. So we didn't have to pull out the hook. That will be followed by questions and answers for all the speakers for 15 minutes. Then we will have half an hour discussion in breakout groups, and I will provide those instructions at that time. And we'll return for a discussion as a whole group, and then we will wrap up and we'll be done by 4, which is exactly what happened yesterday. If you have any comments, questions, suggestions, I urge you to reach out to the data synthesis PIs, all of us here. Our emails are here. And as well as to our NASA OBB program scientist, Dr. Laura Lorenzoni, for comments, support, and anything, that would be excellent. And at the bottom here again is the website for the Arctic Colors. And with that, I will stop sharing and invite the first presentation. Gabby, the floor is yours.
Thank you so much. Can you guys see my screen?
Yes.
Oh, awesome. Okay, my name is Gabriela Negrete-Garcia, and I'm a PhD student at Scripps. And today I will provide a brief overview of the ecological and biogeochemical modeling of the Arctic Ocean. This has been a collaboration with researchers throughout Scripps, NCAR, and now some at GFDL. I will start my talk focusing on the model architecture and sea ice component of the Community Earth System Model version 2. And then I will talk about the Marine Biogeochemistry Library. In short, it's called MARBLE, which is the Ocean Biogeochemistry component of CSM2, which has allowed me to build the new size-based plankton ecological traits model called MARBLE Spectra, which I will talk about briefly. And then lastly, I will show how I have implemented Spectra to understand seasonal changes in plankton communities throughout the Arctic Ocean, something that we're able to do thanks to the larger plankton community incorporated in MARBLE Spectra. The Community Earth System Model is an open source comprehensive model used in simulations of the Earth's past, present, and future climates. It incorporates the ocean, atmosphere, sea ice, and land ice components, and also includes ecosystems, carbon cycle, chemistry, and land ice information. CSM has been at the forefront of national and international efforts to understand and predict the behavior of the Earth's climate, and has been designated to simulate the statistics of climate and not the events of a particular year. The ultimate goal is to better understand the climate system, its interworking and implications. And there are many components within CSM that are very important for the Arctic, but I mostly will focus on the sea ice component and the biogeochemistry component of CSM through this presentation. CSM2 uses the Los Alamos sea ice model size version 5, which is a dynamic thermodynamic model that incorporates a multi-category of ice thickness distribution and multiple vertical ice and snow layers. Each grid cell has 5 different ice thickness categories, and each category has different ice thickness, snow thickness, pond cover, aerosol content, exchange with atmosphere, and exchange with the ocean. And these ice thickness categories provide a means of representing the small scale distribution of ice thickness throughout the model. And the incorporation of size into CSM successfully captures observed declines in average ice extent in all months. You can see this in this figure where it shows the September Northern Hemisphere ice extent in million kilometers squared between 1850 to 2100. And red, there's the observations, the ensemble mean is in black, and the ensemble range is in blue. And this is showing the version 1 CSM large ensemble, which contains 40 ensemble members. When looking at comparing the ensemble mean of the number of open water days throughout the Arctic Ocean between the year 2000 and 2100, it shows the expansion of the open water season over the century with much less sea ice projected by the end of the century. And so the incorporation of size in CSM allows us to have a better have some good projections and predictions of sea ice throughout the year by the end of the century. So now I will talk briefly about the biogeochemistry component of CSM2. MARBLE was designated to be compatible with multiple ocean models and provides a robust platform for researchers to address critical questions related to the impacts of climate variability and change in marine ecosystems. Currently, it has 3 different phytoplankton functional types. One zooplankton incorporates inorganic tracers, detritus, and some phytoplankton community functions such as nitrogen fixation and calcification. MARBLE has some flexibility with respect to the configuration of its ecosystem, supporting simulation of arbitrary number of zooplankton and phytoplankton functional types. So in brief, MARBLE is the ocean biogeochemistry component of CSM2. It has a flexible plankton functional type model with a modular architecture supporting portability across ocean circulation models. And something to note is that CSM2 CMIP6 simulations contain significant biogeochemical biases linked to underlying physics. But MARBLE has allowed me to create the new size-based plankton ecological traits model called MARBLE Spectra, which builds on other ecosystem models. However, it is unique in that it harmonizes the strengths of plankton functional types crucial for capturing large-scale biogeochemical cycles with the strengths of size-based models aimed at representing the richness of plankton food webs and trophic dynamics while making it lean enough to be able to run for century-scale simulations. MARBLE Spectra simulates important marine ecosystem dynamics such as phenology, geography, and trophic dynamics, as well as the couple cycles of carbon, nitrogen, phosphorus, iron, silicon, and oxygen. It's currently a handcast model from 1948 to 2009, where phytoplankton are dependent on growth, predation, and loss, and zooplankton on ingestion, predation, and loss. And this is a little schematic of how the ecosystem looks like. We have 9 different phytoplankton groups, which we were able to increase the number of phytoplankton through the use of allometric scaling techniques. The smallest phytoplankton are the picoplankton. Then we have the diazothropes, which are the nitrogen fixers. We have 4 different mixed phytoplankton groups, and within them, there's the implicit calcifiers. And then we have 3 different diatoms, which are the silicifiers. They all take in nutrients and are eaten by zooplankton. For the zooplankton, the smallest 2 are the microzooplankton, and the largest 4 are the mesozooplankton. They feed on phytoplankton, but also on other zooplankton. And once they are eaten or die, they're converted into the particular organic matter, dissolved organic matter, and dissolved inorganic matter pools. There's many allometric relationships that I have used to create this model, but I'm only going to show one as an example. We used empirical observations to be able to scale these phytoplankton with size. And here I'm showing the half saturation constants for nitrate and log base 10 parameter value with the log base 10 spherical square diameter. And here are the different functional types in different blue colors, and the different sizes are there. This half saturation constants tend to increase with increasing cell size, such that smaller cells have higher rates of nutrient uptake per unit biomass, and therefore lower half saturation constants due to their higher surface to volume ratios compared to larger cells. And the only difference here is that, or exception, is that diazotrophs are not very efficient at inorganic nutrient uptake, and they often occur as large colonies where their surface to volume considerations imply higher half saturation constants relative to small phytoplankton and diatom groups. For zooplankton ingestion rates are calculated as a function of prey carbon concentrations using the Rawlins type 2 function, which depends on the maximum ingestion rate and half saturation coefficient, where the maximum ingestion rates decrease with increasing body mass, indicating a slowdown in the metabolism. And the half saturation coefficient is independent of body size and regulates ingestion efficiency at low biomass. And these ingestion rates are also modified by the feeding kernel, which represents the probability of a given predator ingesting prey of a particular size. And this probability is represented as a modified Laplacian distribution centered at a mean predator to prey ratio for all zooplankton predators, which shows the preferred size of prey for all zooplankton, with deviations from this ratio decreasing the amount of grazing. And you can see that the different zooplankton have a slightly different probability or range of prey, and the larger zooplankton would have a greater range compared to smaller ones. There are many comparisons that I have made, but I made some results in how the general community of phytoplankton scales with size. And this is comparing it with satellite-derived size classes from Hirata et al. 2011, where the smaller phytoplankton are seen to be dominating oligotrophic regions of the ocean, which makes sense since these regions tend to have lower nutrient concentrations, and that is consistent with satellite-derived observations from Hirata. The medium phytoplankton dominate coastal and productive regions of the ocean, which tend to have higher nutrient concentrations, and that is also something that has been observed. And the large phytoplankton dominate polar regions, which is consistent.
Gabby, are you there? Is anybody else there?
We have
Yep,
seen
I can
that.
still hear you.
Oh, sorry. Sorry, can you hear me now?
Yes, we can hear you now.
Oh, okay, sorry. Generation times tend to increase with increasing cell size. And you can see this by in the y-axis, there's, I'm showing log base 10 generation time and days, and then the x-axis log base 10 mass. And then blue is phytoplankton and orange zooplankton, and generation times tend to increase with increasing size in the model. For the zooplankton to phytoplankton biomass ratios, overall, you can see that these ratios tend to be at 1 or below 1 throughout the globe, and they tend to decrease with a biomass gradient. And this is consistent with observations seen that in locations with higher biomass, the community tends to be more dominated by phytoplankton compared to zooplankton. And lastly, for the zooplankton mean trophic level, we tend to see higher trophic levels in oligotrophic regions of the ocean, which is consistent with what we would expect since these regions tend to be dominated by smaller phytoplankton, leading to longer food chains and a less efficient transfer of carbon into higher trophic levels. So lastly, I wanted to give a little summary of how I've used spectra to understand seasonal changes in plankton communities throughout the Arctic Ocean. And some of the main questions I want to ask and answer in this presentation are how does the distribution and composition of Arctic phytoplankton communities change over the year, and what are the regional differences, and which size classes are limited by which factors at what times of the year, and how does this vary in space. And I have done this by separating the Arctic into 10 different regions of the ocean, but for simplicity, I will only look at the Central Arctic, the Laptip Sea, which is an interior shelf ecosystem, the Nordic Sea, which is influenced by Atlantic inflow, and the Chukchi Sea, which is influenced by Pacific inflow. And here I'm showing biomass of phytoplankton in red and zooplankton in blue. And overall, in every single region, you can see that phytoplankton blooms are followed by zooplankton's peaks in biomass in every region, but there's some regional differences in phenology and also in magnitude. So you can see that the smallest magnitude is seen in the Laptip Sea, which is an interior shelf ecosystem, and the highest biomass is seen in the Nordic Sea due to the Atlantic inflow. I also wanted to see how the community structure changes in the Arctic throughout the year by plotting the slope of the size abundance relationship, which would show that a more negative slope would show a higher contribution of smaller cells in the ecosystem, and a less negative slope would show a higher contribution of larger cells. And so you can see that there's a higher dominance of smaller cells during the summer, which could be due to the depletion of nutrients after the productive period. And this can be better explained by plotting the slope of the size abundance relationship in every region of the Arctic throughout the year with nitrate concentrations. And you can see that the more negative slopes tend to be consistent with lower nutrient concentrations during the summer, showing that the seasonal size structure is driven by nitrate availability. And lastly, to ask which size classes are limited by which factors at what times of the year, and how does this vary in space, I have plotted the growth limitation, the most limiting factor of growth of different phytoplankton throughout the year, where in yellow, I'm showing light limitation, in blue, temperature limitation, and in green, nutrient limitation. And you can see that picoplankton are limited by light and temperature throughout the year, while large diatoms tend to be limited by nutrients during the summer. And so in summary, CSM2 is a powerful tool for studying dynamics in Arctic Ocean ecosystems due to changes in the coupled climate system. It has many existing simulations available to research community, and some of them include large sample simulations, over 100 simulations for 1850 to 2100. MARBLE provides a flexible framework to incorporate more plankton types in ecological models. In MARBLE, spectra can simulate seasonal and regional changes in plankton phenology and succession, and their roles in ecosystem functioning and biochemical processes. And the incorporation of MARBLE spectra and CSM2 enables mechanistic projections of how plankton communities are responding to seasonal and interannual changes in the environment, as well as how they might respond to future environmental change. Thanks.
Wonderful, Gabby. Thank you so much. Questions will be at the end. Joel, your turn.
All right. Thank you. All right. I really appreciate the opportunity to speak to the meeting. I think there's a lot of great synergies between what we're trying to do on the Interface project and what is being proposed for Arctic Colors. Interface is a DOE project. It stands for Interdisciplinary Research for Arctic Coastal Environments. And so today, I just plan to give you a fairly high overview of the various activities we're engaged in and the research we have. So to start with, the project is 2 years old. We're going into our 3rd year, and it was designed around what is very similar to the Arctic Colors, a systems approach with the recognition that to understand coastal change, really need to look at the interaction between land and ocean and the engagement at the coast itself. And our project, which is sort of graphically shown on the right, is centered around Earth system modeling, but with a hierarchy of models with a range of particular topic focuses, from coastal erosion to sea ice to shipping to biogeochemistry to natural resources. And to do that, we need to have the coupling of the land to the ocean, and then the recognition of, along with other DOE coastal efforts, which has started recently, the Arctic is unique from all other coastal regions, particularly with the presence of sea ice, permafrost, and its sensitivity to global drivers and feedbacks. So to give you a little bit of background, overall, a very wordy slide is, as I mentioned, we're funded by the Department of Energy. Our funding comes from the Earth and Environmental Systems Sciences Division, which used to be the Climate and Environmental Science Division. And out of that, we're a somewhat unique program in that we're funded out of 3 different program areas in the Earth system modeling area and the data management side run by Jay Nilo. And so we receive funding on the regional and global model analysis, model development, which is where the Earth Energy Exascale Earth System Model, E3SM, model development really lives, and then multi-sector dynamics run by Bob Valerio, which is really focused on the human economic dimensions. And I won't talk as much about that today, but it's a critical component of our effort. Interface is comprised of 5 institutions presently: Los Alamos, Oak Ridge, Pacific Northwest National Labs, Sandia National Labs, University of Alaska Fairbanks. NEMCAR will hopefully be joining in this coming year as we get the contract issues worked out. We designed the project around 4 common Earth science areas: sea ice and ocean dynamics, permafrost hydrology, coastal change, and marine biogeochemistry. And then on the human multi-sector dynamic side, we've been working to integrate that with transportation, resource development, settlement, and marine ecology. And so broadly, we motivated our project into our various program areas, but also with cross-cutting themes that were highlighted by these 3 themes of uncertainty, really looking at the predictability of future humans, environmental systems and interactions of complex human systems. And that's being represented by the multi-ensemble run and the level of variability just in Earth system models. The next, as a topic that was talked about a lot yesterday, is seasonality, and really highlighting the fact that so many components of the Arctic are strongly tied to seasonal rhythms and seasonal dynamics. And as those shift and change, their interactions will be critically important to investigate. And finally, we have an area we're calling typologies, which is really an effort to characterize the Arctic system, particularly the Arctic coast, based off its physical and environmental attributes and also its propensity for change. And that, as I'll highlight later, is used to both look for representation, model development, and model testing. And so the next aspect of it is I will go through some of our particular model development and focus areas that we have related to modeling. And so related to E3SM, one of the key areas that we've focused on is ocean and sea ice dynamics. And in the upper right, what I'm showing is the mesh that we're using for our simulations in E3SM, which is a hybrid mesh of multi-resolution with increased focus across the entire Arctic. We go from a 60-kilometer globally down to a 14-kilometer resolution in the Arctic. And that allows us not only to resolve shipping lanes, but also allows us to begin to look at sea ice, in particularly the coastal regions. We've been working on incorporating WaveWatch 3 into E3SM for a global model, and that's represented by the second figure that we do have it running in a global configuration to work at the poles, which will be critical for examining wave and sea ice interactions. We've been working on developing a new mixing for the ocean that is better, works better in the Arctic, is more appropriate, and that's the assumed distribution higher order closure. And then we're incorporating the land-fast ice from the sea ice repository into E3SM to be able to examine coastal constraints. And on all these slides at the bottom, I'll list the team members who are working on various components of the project and their institution association. And so please feel free to follow up with me or look them up if you have particular questions, but I'll definitely get you in touch with people if you have follow-up questions on specific aspects. Next, another component that's being worked on is the biogeochemistry, and this is really being led by Nicole Jeffrey out of Los Alamos, and she's working on developing a new benthic active layer diagenesis submodel to be incorporated into E3SM. And the bottom right shows the schematic of the processes that she's been developing into it, and the upper panel shows a validation effort that she's been working on using data from the Arkona Basin. Also related to biogeochemistry is an active effort led by our biogeochemistry team, particularly our collaborators at UAF, of data compilation and model analysis with a particular focus on primary productivity in the Arctic. And that's been leading to work on the human dimension side of working with people at the International Arctic Research Center of understanding the coupling of primary productivity changes and other environmental indices to look at food security, so such as timing of open water, availability, and distance that people would have to travel to access marine resources. Also, looking at outside of the E3SM framework, we're working on what has been described as a hierarchy of models, so really looking at high resolution specialized models to begin to investigate questions of coastal erosion and flooding. And our two main modeling tools for this are the Arctic Coastal Erosion Model developed at Sandia National Lab, which I know a number of people here have collaborated on some of the field efforts to help parameterize that model. And then the use of the Advanced Terrestrial Simulator model of for looking at coastal flooding. And on the right, we're showing different flooding scenarios that we're looking at to understand the impact of inundation on low-lying permafrost and permafrost thaw. A key component of the land side that we're working on is the watershed hydrology and multi-scale modeling. And this is an effort really being led by Ethan Kuhn out of Oak Ridge. And once again, this is using the Advanced Terrestrial Simulator to be able to work at watershed scale simulations. This is a model that has permafrost, but also has subsurface and surface water flow, and runs on high performance computing network. And so it has a lot of advantages over system models with a simple column based with lateral transport. And we're pioneering an effort to try to use a simplified representation of hill slopes that scale up to an entire watershed, which is then coupled to the routing model within E3SM, which has the capability to transport heat, sediment, and nutrients as well. And so this is being developed in collaboration. A lot of the data that's supporting this is coming from collaborators at UT Austin and the work across the LTER work on the North Slope of Alaska. We also have, talking about typologies, this is an example of the work being done by Ben Jones and Anastasia Plores. What I'm showing you is clustering along the Arctic coastline. And this is based not only on historical erosion rates, but the physical properties of the coastline, including things like ice content, geomorphology, soil properties, coastline orientation, sheltering of the coast. And in order to classify the coast in unique areas to understand the variations and patterns along the coast, this represents both a data synthesis effort, but also gets at representativeness and transferability of the individual coastal regions to other areas. It helps us understand where there are data gaps and whether or not portions of the coast which are highly studied represent the larger portion of the coast, but also provides us a pathway for model development and model testing, and eventually for upscaling parameterization of erosional fluxes potentially into Earth system models. This is what I'm showing you here is the onshore typologies. We have a complementary effort looking at offshore typologies, which look at the bathymetry, wave climates, and other governing equations which drive coastal erosion and coastal change. And we're looking at synthesizing those together. Next, we have a component of our effort, which is of value, is a data component. Our project is funded out of the data management program, and that has multiple components, including efforts at UAF to stand up a unique data archive, but also an effort to go and build out data sets which are critical for interface science. And this is an example of related to watershed hydrology, an effort to go and synthesize, find, and curate data sets that will be needed for the hydrologic and land modeling. That includes snow, river discharge, soil temperature, thaw depth, and soil properties. And not only is this going and finding data that's in existing archives, but most importantly, finding non-archived data sets, such as material that are old reports, publications that were never electronically incorporated, and also even in log books of researchers who've worked at UAF and worked in Alaska for a long term. And so this is an effort to bring together data sets that allow us to extend both the temporal range and the geographic coverage of our ability to work on our modeling and to focus on it. And so finally, to wrap up, our other and future efforts, which I'm happy to talk in more detail about, are the Arctic Data Collaborative, which is being led out of UAF. This is really looking at a particular focus on finding and curating data sets which are of great interest to stakeholders and making them available for stakeholders in Alaska, as opposed to duplicating archiving efforts on NSF or even DOE. Another data set that we've been working on curating, we're almost done, is a sea ice data set. I think Hayo Aiken might touch on that tomorrow when he's talking about data at UAF. Another key component of our efforts within the DOE framework is to develop model benchmarking tools. And this is focused through the ILAM effort from DOE. And we focused on predictions of river discharges. And so assembling data sets to allow us to interrogate models, but also building in new tools into ILAM that allow us to look at both watershed and point-based measurements to understand how well models are capturing river fluxes and particular aspects of river discharge that are critical for understanding the interaction between land and the ocean set. We've also worked on a major effort to compile and analyze Pan-Arctic river data sets that go beyond just the GROW data set, but across larger Arctic, particularly with a focus on smaller watersheds, and also a focus on human influence and dams across the region to understand the influence between natural and human-driven changes. Finally, we have researchers looking at examination of base flow trends and active layer changes. And then a big component of our project, which I didn't touch on a whole lot, is coupling it to the human system and understanding feedbacks between the global economic drivers and human change in the Arctic. And that includes shipping, pipelines, resource development, and drivers for community impacts to communities. And so an ongoing effort that we have, and this is one of the reasons why I'm very thankful to be participating in this effort, is identification and development of collaborations across the Arctic research community. And a number of you have had conversations with us in the past. Some are working with us in terms of data and modeling efforts, and we'd really like to build that out. Thank you very much.
Thank you. Wonderful. It's wonderful to hear. Please either put your questions in the chat, and then we will come back to those after the 4 presentations are ready. And so, Mike, if you're ready to go, please do so.
Okay. See my screen?
Yes, we do. Or I do.
All right. Thank you. Really happy to be here with you all today to talk about some freshwater and dissolved organic carbon modeling. First off, you may notice from the title, I'm not just speaking about the North Slope here, but I'm also including some modeling work that encompasses the Western Arctic. And for that purpose, we're looking at the land areas that drain between the outlets of the Yukon and Mackenzie rivers, including those. So essentially the Arctic colors domain. And much of this work that I'll be describing is recently published in a journal article in Journal of Geophysical Research. Before talking about the modeling itself, it's important to mention the key data sets that we use to calibrate and validate the models. So as we heard yesterday, the Arctic Great Rivers Observatory is a key data set for the large rivers. So for the Yukon and the Mackenzie, as we heard yesterday, there are enough a long enough time series and frequent enough sampling to give us baselines of freshwater and dissolved organic carbon exports from the land to the ocean for those large rivers. But of course, for the medium and the smaller size rivers, particularly the smaller rivers throughout the Pan-Arctic, we need other data sets. So for observed river discharge data, we have, again, Mackenzie, the Yukon on the North Slope, the Colville River, the Koparik, the Sag. There are USGS data that gives us freshwater fluxes for those rivers. But for many of the smaller rivers, the data are really spotty. For DOC concentration, the Mackenzie, the Yukon, the Koparik, select North Slope rivers. I'm not certain that there are more frequent samples now than there were earlier in the decade or the past several years. That's open for discussion. Also want to mention that there's been some efforts to do some modeling of dissolved organic carbon. The terrestrial ecosystem model, Kicklighter et al. 2013 presented those estimates. So there has been some work in that area to date. As we heard yesterday, there are data that we can use from sample data, we can derive empirical estimates from river sampling. Just want to speak to the DOC concentrations in the upper panels. So for the DOC concentration, we can use these samples to give us estimates across watersheds across the North Slope of Alaska. DOC concentration is essentially a function of watershed slope. So those variability in soil organic matter helps to explain these slope DOM linkages that we see from these data. The numerical modeling that I'm doing is leveraging the permafrost water balance model. So that model uses parameterizations for soil texture, rooting zone depth, soil organic carbon content, which is shown in the lower right there from the NCSCD data set. The model typically ingests meteorological data from reanalysis. We can also force the model with observations, for example, from Ukon-FVCOM Airport that I'll speak to in a moment. The model incorporates all the major elements of the Arctic water cycle. We've got a tracking of snow accumulation and melt, a physically based soil thermal module, soil water. The model outputs then, the runoff can be ingested into a routing model, which will give us then freshwater export and DOC export based on the latest updates to the model. And in the lower left there, you see a schematic showing that the model accounts for properties of organic soils, mineral soils. It's got an active layer component, physically based soil freeze thaw, surface pool. And of course, to do terrestrial hydrology, we need to use essentially, if you will, digital river networks. And in the upper right there is showing the depiction of the digital river network for the area of Northwest Alaska that's draining to Elson Lagoon. We've recently established a new 1 kilometer river network for the North Slope of Alaska. That's 182,000 square kilometers. We've got defined from this network, then we can define at 1 kilometer resolution the watersheds that are draining to different areas of the North Slope. So as you can see here, the map of Elson Lagoon there, that's an area that's larger than the Elson Lagoon domain that I've defined here on the left. The one on the right there is including Admiralty Bay. We've got also the drainage to Simpson Lagoon, and farther to the east, Jago Lagoon, Kaktovik Lagoon, the Canning River. We've got defined all these different watersheds that will give us at 1 kilometer resolution the terrestrial hydrology, the drainage, freshwater, and dissolved organic carbon fluxes. The permafrost water balance model was recently updated to include DOC process representations. So DOC processing, DOC production is essentially a function of soil organic matter content. And also there are parameterizations for the influences of soil temperature and soil moisture. You increase temperature, you increase moisture. Shown on the right there, we get increased DOC production. We've got decomposition also of the DOC pool in the soil water pool. And that's also a function of the amount of soil water, air temperature, soil moisture again. The soil water DOC pool is updated each day then, and the leaching and loading to the stream network, that is a function of the amount of soil water DOC and occurs whenever we have a runoff event, which is Q. And so as I'm showing here on the upper right, in the upper right here, DOC production, for example, in milligrams of carbon, increases with soil temperature and increases with the amount of soil water fraction, the amount of soil water we have. And the DOC leaching then is greater when we have more soil water DOC storage and when the DOC concentration is higher. That gives us more leachate that is then loaded to the streams. So then in order to get a good estimate of the dissolved organic carbon exports and concentrations, we need to have a good approximation of the hydrology. So this is runoff. This is validation of the simulated freshwater export for the Colville, Kuparuk, Mackenzie, and Yukon. Showing in there simulated in blue, observed runoff in red. And what we can see here is that at monthly climatology, the monthly time step, the model does extremely well at getting the spring freshet for the Colville and the Kuparuk. Now for the Kuparuk there, if we add together the simulated in May and June, we then match very closely the amount of runoff that's occurring in June. So the model is a little bit early in the snowmelt pulse for the Kuparuk, but nonetheless, percent differences are within about 4% for the Colville, Mackenzie, and Yukon, about 11% for the Kuparuk. So the model is capturing the amount of freshwater export that's occurring, at least particularly for when we look at a monthly time step. Then looking at simulated runoff DOC loading and concentration for 4 focus basins at a daily time step. So these traces here are at a daily time step. And what we can see here is in the red, the DOC concentration in the runoff, as you can see here, it is highest in the Colville, the Kuparuk, and close to, for the Colville and the Kuparuk, the highest DOC concentrations occur very close to the time of peak runoff. And that may be counterintuitive, but that's what's happening out in the real world. We're seeing that the DOC concentrations in the discharge is highest at the time of peak runoff, and the model captures that. And of course, then the loadings in black, then the mass loadings are then much higher during that time in the spring freshet. That's when we get the large export of dissolved organic carbon. For the Mackenzie and the Yukon, the timings aren't quite as close as they are for the North Slope, but still we're getting that pattern, particularly there, if you can see for the Yukon, much, much higher DOC exports at the time of the peak runoff. And I should mention here as well, we like to understand whether these higher concentrations, particularly in the Colville, and also to some extent in the Kuparuk in the fall, we don't have any measurements then in late August, September, and maybe early October to validate these, arguing for the need for more data, observed data. If we look then at the Yukon and the Mackenzie, we can see that the Yukon and Mackenzie, the simulated loadings here in the black bars are slightly higher than the observed, and these are observed loadings, or the observed exports, I should say, from a number of studies. If we compare the simulated to the observed, we see that the simulated matches well with the observed at the seasonal time step, so we can get seasonal exports from these rivers. The loading should be higher than the exports because losses are assumed to occur in transit to the coast. For 6 of the 8 comparisons, we're within about 25%. So that's looking good. We can essentially get these seasonal exports. But again, this is loadings. This is not actual exports to the coast because we have to take into account losses in transit. If we look at DOC concentration for the North Slope rivers, we can see that the modeling captures the east-west gradient in DOC concentrations that we heard about yesterday. So the simulated DOC concentrations for the North Slope rivers matches well the observations that are derived from the empirical estimates that we talked about earlier. That gives us DOC yield for the whole Western Arctic. Then DOC yield in grams of carbon per square meter per year. Let me just skip ahead. The estimates for the Yukon and the Mackenzie are close to those estimates that we get from Holmes et al. Based on the sampling of the rivers. Slightly higher, about 15% higher, that's the losses in transit. So then that gives us for all of the major regions throughout the Western Arctic, the Yukon to Mackenzie region, Mackenzie Basin, Yukon, Western Alaska, and North Slope of Alaska, monthly loadings to the rivers. The monthly loadings peak in May, the exports peak in June, and transit time accounts for that. We get a roughly equivalent loadings to the Beaufort Sea and to the Chukchi and the Chukchi Sea and the Bering Sea. So roughly equivalent loadings for the rivers going to the west and to the north. If we look at increasing freshwater and DOC exports, so another study that was recently published, we took the model and looked at exports, DOC export and freshwater exports to the area draining to Elson Lagoon in Northwest Alaska. And long story short, there has been increases in freshwater export and DOC export based on the modeling over the period 1981 to 2010. Particularly, you have a large anomaly here in 2019, a large freshwater anomaly, and much increased super permafrost subsurface DOC export here, particularly in September and in October. Why is that? Well, we're having really large increases, strong increases, significant in air temperature, particularly in autumn, and in precipitation. This is an influence of the sea ice losses manifesting in the area in Northwest Alaska. We could see the soil moisture vertical profile much deeper in 2017 and 2019 versus the early 1980s. So we've got a larger volume of thawed soil, more production, warmer soils, more productivity, leading to increased DOC export along with the freshwater export. So for current model estimates and plans, we've got major rivers covered, daily time step 25-kilometer resolution for the Yukon to Mackenzie region, 1-kilometer resolution for Elson Lagoon. In progress, we're modeling the select North Slope watersheds at 1-kilometer resolution using DayMet, forcings, and Error 5. And we have plans for future simulations for the full Western Arctic domain using GCM air temperature and precipitation from the SNAP program. We'd like to do some model updates with river wave flow propagation and lateral subsurface flows, surface water to stream dynamics as well. So in summary, the process modeling is providing freshwater discharge and DOC loadings within about 10% for freshwater and about 15–20% for the DOC. Higher resolution needed for the smaller rivers. The modeling captures this east-west gradient in the DOC export concentrations. Warming climate, hydrological cycle intensification, and permafrost degradation have altered freshwater and DOC flows to Elson Lagoon. We definitely need additional river samples during the shoulder seasons, particularly late summer and autumn for the smaller rivers, and accounting for surface to stream water dynamics, local processes, and last but not least, river in-stream processing is a big missing component here. Thank you.
Thank you, Mike. And last but not least, Blake, might you be ready? Please remember, keep posting your questions. I'm keeping track of them, and we'll address them after all the talks are done.
Yeah, let me pull up my screen real quick. Okay. And then can you see the
Yes.
presentation? Okay,
Yes.
great.
Excellent.
So
Thanks.
Yeah, so thanks everybody. My name is Blake Clark. I'm an associate scientist with USRA guest star at Goddard Space Flight Center. And I'm happy to present some work today that I started as a NASA postdoctoral fellow, building a model of the Yukon River Delta to understand and better estimate organic carbon export and processing in Arctic deltas, plumes, and the coastal ocean. And I'll talk a little bit about this model, and then also talk about some work that we have ongoing on the North Slope, and then also sort of end with some questions and challenges I think that we have as a community. So the first thing I wanted to show was the actual model that I use that was developed by Chongsheng Chen out of UMass Dartmouth, which is the FVCOM hydrodynamic model. So they've actually implemented this model with 3 different resolutions for the entire Arctic Ocean. So here I'm showing an animation of sea ice on the along the Alaskan coastline. And from this study, they show that the model resolution, which in the middle column, resolution is increasing as you go down. So it goes from about 8 kilometers in the upper box to 500 meters in the lower box. That, of course, with different model resolutions, you get pretty starkly different results. And it just so happens that comparisons with the observations for both temperature and salinity, as well as velocity shown on the right through the Bering Strait, the higher resolution models tend to perform better. But of course, there's the computational cost with that, where the highest resolution implementation of this model, it was about over 1 million grid cells and took about 600 hours or 600 wall clock hours to run on 500 CPUs about. So there's always this computational cost and this balance between having, realism and high resolution and high accuracy and actual feasibility, technical feasibility. But I think that the continued push up against what's possible in terms of computational ability is necessary to resolve the coastal Arctic. So here I'm showing an image from NASA Worldview. It's a true color composite image from June 12th of 2019, when we were actually out there sampling. And this video on the right is from a sampling trip that we took. And you'll see that there's these really strong frontal features and these billows that occur within the plume. And you can even see them in real time, where you have these huge lines of mixing that are occurring, where foam develops, and you could have really strong differences in physical water properties and presumptively biogeochemical properties in spans of tens of meters. So not only do we need to have this really high spatial resolution, but we also have to include these complex processes, further putting a demand on the computational ability that we have. But luckily, we have government agencies such as the Department of Energy and NASA that have continued to build out these supercomputing platforms that give us, as Earth scientists, the technical capability to sort of push the bounds of what's possible. And so today, really what I want to talk about is the model that we've developed for the Yukon River, the Yukon-FVCOM hydrodynamic model. And so first, I'll introduce the hydrodynamic simulation, which is mostly looking at the physics. And then I'll talk more about carbon cycling and looking at carbon fluxes within the Yukon Delta. So on the left is the model domain, which starts at Pilot Station and runs through the Delta, including all of the Delta channels, or the vast majority of them, and then out towards the coastal ocean. And I'm showing a transect, actually the transect that we conducted as part of this, the NASA Remote Sensing of Water Quality project in 2019, with this blue or cyan line. And then the red dots, or red diamonds, represent CTD profiles that have been collected routinely by the Alaska Department of Fish and Game as part of their annual red king crab survey in August. And you'll see that the model structure on the right has this triangular stretching form, so that it allows for it to resolve these very complex bathymetric and geomorphic features. And our model resolution stretches from about 2.5 kilometers at the boundary in the deepest waters to less than 100 meters up to about as low as 30 meters in some of the shallow waters, and then up towards Pilot Station. And so for this model, we've run it for 7 years from April 1 through September 30th for 2004 and 2005 and 2015 through 2019. And the statistical analysis I'll show next, we start the model analysis on May 15th because we don't have a representation of dynamic sea ice, although the capability is possible, and we hope to implement that in the future. And for this model, each run takes about 20 wall clock hours using 560 CPUs on the NCCS Discover supercomputer. So here's showing a model data comparison. And of course, we want to have the physics properly represented to get a good biogeochemical representation and carbon cycle representation. So on the left, I'm showing a representative sea surface temperature comparison of Modus Aqua and the actual Yukon-FVCOM sea surface temperature. And we did comparisons for all of the months across all years, and in terms of the average SST, of course, because it's quite cloudy in the region, so you can't really compare individual days or images. And we also compared the CTD profiles shown on the bottom right using these target diagrams, where the closer you are to the middle, essentially is better. And in terms of temperature, the model has a slight cold bias, especially deeper, which indicates that there's some physical process that we're not quite representing in terms of how heat is being transferred through the water column or from the open boundary. But in terms of salinity and stratification, the model does quite well, and there's very little bias. And so we feel pretty good about the bulk physics that we're representing within the plume, which are going to be density driven, and where the density is going to be primarily determined by these really strong salinity gradients. So we analyzed 7 years of varying hydrology and to better understand these controls on river plume dynamics. And this is from a paper that myself and Antonio Menino have submitted. It's in review in Frontiers in Marine Science. So on the left, I'm showing the hydrographs from Pilot Station from when the record started back in 1977. And you'll see that, in 2005, had the highest discharge, which actually occurred quite early. 2016 had the lowest discharge and had this sort of odd peak that occurred in late August. But there's lots of variability, and there's lots of variability across all of the years that we included in our study. But what we wanted to look at was the effect of wind as sort of this secondary control on plume structure. And what we found was that southerly winds, or downwelling winds in the left column, enhance the mean northward flow. And this is what we would expect based off of other studies, such as from the Columbia River and other modeling studies dating back to the early 2000s. So you have this enhancement of the mean flow with downwelling winds, or winds that are blowing to the north, and winds that are blowing to the south, or upwelling winds. You have this sort of reversal of flow and in an enhancement of stratification and a broadening of the river plume. So wind can have a really strong effect on the river plume distribution. And we hope to use this knowledge moving forward when we conduct sampling, like coming up in this next year, so that we can better target where we should go and when we should go there based off of the previous recent wind history. Because it appears that although the first order control is going to be the fresh water and when the freshet occurs, that really across years the freshet is relatively consistent, and it's these other factors such as wind that are going to determine a lot of the short-term variability. So that's just a basic description of the physics. But really what we're interested in is the biogeochemistry and carbon cycle. And so we have developed this model, and this actually goes back to work done in the Chesapeake Bay over 30 years ago. And we developed this model to focus on how organic carbon is cycled at the land-ocean interface. So on the left, I'm showing two schematics with the spring freshet in the upper panel, and then the late summer in the lower panel, and the dominant processes that we're representing. And it's sort of summarized on the right. And we do include a full sediment flux and diagenesis model as well. Importantly, we have 6 DOC pools, and we include photochemical degradation of CDOM, and we have hyperspectral light attenuation and water leaving gradients, phytoplankton growth and death, of course, and particulate organic matter sinking and resuspension. And the model actually does pretty well at capturing DOC and POC, which we measured in this research cruise in 2019. So these are some of the only measurements that we have in the region to validate the model for. And they were all collected within sort of a span of 4 weeks in June. So of course, modelers always say we want more data. Well, we do need more data to more accurately assess whether the model captures the interseasonal variability. But it appears that at least during this spring freshet period that we're doing quite well at modeling the bulk DOC and POC concentration. So of course, then we wanted to look at fluxes. And with this model, we have the ability to extract information across transects and actually calculate the exact flux, the exact integrated flux through time and space across these areas shown here in orange. So pilot station, of course, is where the water's coming in, and we're loading with estimated concentrations using the Arctic GRO data set and LODES software. And then we have these various river channels along through the delta, and then a plume transect that is roughly approximated with the 10-meter isobath. And so here are the results of the fluxes. So on the left, on the upper left, we see the flux of DOC, and the upper right, we see the flux of POC. And then on the bottom panel are sort of bar plots showing the distribution among these different classes of POC and DOC. So the total flux of organic carbon is 1.6 teragrams, which 1 teragram of which is DOC and 0.6 teragrams of which is POC. And 80% of the organic carbon goes through the south mouth. And being there, that's not surprising, and sort of understanding the geomorphology of the delta, that's not so surprising. But the 80% number is quite higher than I would have expected initially. And what's also very interesting is we've calculated that about 38% of the organic carbon that's leaving the delta exits across that plume transect. And 49% of that is DOC, with 22% of that being POC. And the distribution of the organic matter changes as well in terms of the composition or the quality, where of course what's going out of the plume transect is going to be more marine in nature. And that's based off of the optical properties as well as the sources within the model itself. So the last thing I want to talk about is this new project we have through the NASA Ocean Biology and Biogeochemistry, and it's led by Wes Moses from NRL, where we're building out a new system in the Beaufort Sea around Prudhoe Bay, which including the Colville, Kuparuk, and Sag rivers. And so we've built this initial model that has about 300,000 triangular elements, and you can see where it is with geographic relation to the Yukon model. And we're in initial simulations, and things look promising so far. And of course, with the LTER data, we have a good amount of data to validate physics, including velocity, using the mooring data that's available. So that's really useful. And also, we want to try to build out eventually to this sort of hypothetical domain that I plotted here on the left that includes a large portion of the Arctic Ocean, including the Bering Strait. And it's extremely computationally intensive to do that, because of course we need to have high resolution to capture these coastal regions up into the rivers. But to do that, then we're looking at having, maybe 5 to 10 to 20 million elements. And even building the model grid, which I haven't had success at yet, is quite computationally challenging. Just actually assembling the elements into this triangular mesh structure. But I'm hopeful that we can do it, and I'm hoping that, you know, with collaboration we can include other processes and inputs such as Mike Rawlins' model for DOC inputs in some of these unmonitored systems. And I'm hopeful that we can actually sort of build out a really high resolution system for the coastal Arctic. And finally, I know I'm a little bit going over here, but there's more carbon is to come in these models. So we have a carbon cycle science project through NASA that got funded, that we're starting up early next year, where we're going to include the inorganic carbonate system into the model that we've already built. And luckily, a lot of this work has already been done by Laura Bianucci, who was at she was at the PNNL, Department of Energy. So it's well tested and established, and we're looking forward to that. And of course, there's measurements from Arctic GRO to actually force the model from pilot station. The other thing that we're looking at doing, and that we're going to do, is build out the planktonic food web to include more things such as mixotrophy and other types of diatom functional groups that are going to more accurately represent the phytoplankton community within these coastal systems. And that's shown here by this diagram from Kellogg et al. 2019. And so I'm looking forward to continuing this work and hopefully have more to show in the future. And just wanted to thank all these folks who have worked with. A lot of them, of course, are part of the Arctic Colors community. And I hope to work with more of the folks on this call in the future. And I guess I'll leave with this slide, just thinking about main challenges and opportunities moving forward. So I think linking models, of course, is quite challenging, and across these model types. So we've heard about, of course, sea ice, erosion, planktonic food webs, higher trophic levels such as like food fish or benthic grazers, and of course the human element that's going to impact these coastal systems. And also acquiring and knowing how to use data is challenging, especially knowing what data is good, and that was touched on quite a bit yesterday. And then, of course, what we talked about, scaling up, including the sea ice biogeochemical properties just beyond the physics. And then I think building the technical capacity and working with engineers so that we can do more earth science and less computer science and have more efficient models that work really, really well on high performance computers. And I think that frees us up to do hopefully some really cutting edge research into the future. And with that, thank you.
All right. I want to thank all of our speakers, Gabby, Joel, and Mike, and Blake, who almost got the hook, but he had a really good summary slide with issues to discuss. And so now, over the next 10 minutes or so, is the opportunity to ask all speakers questions. And there are two already in the chat. And the first one is to Gabby, and I know she's read the question already, so I'm going to let her address that question. It's a conversation.
Yeah, so thank you, Lee, for this question. I didn't speak much about the benthic or how nutrients are modeled in spectra, but it does, spectra does simulate nutrient concentrations throughout the water column, as well as and then it computes denitrification losses of material to the seafloor, and I just didn't speak much about it in the presentation. And also, spectra was like was created initially just to, or was created to be a global model. And so that's why we have incorporated diacetrophs in the model, but we don't simulate them in the Arctic. And so they're there because they will be useful for whoever wants to use them for, you know, more of tropical regions, but yeah, they're not simulated in the Arctic.
Okay, thanks. Thanks very much.
Thank you.
So I have a question for you, Gabby, with respect to this, and that is, what is the current footprint, if you wish, the spatial resolution at which marble or spectra are operating? How far? I understand that, no, the higher the resolution, the more costly it is, but if one reduces the region for which one is applying it, then one can enhance the spatial resolution.
Yes, so the resolution is still 1 degree by 1 degree, like similar to marble. So yeah, it is very similar to the CSM and the marble component.
Thank you. The next question we have is one that Robert Friend poses to everyone, not only the modelers who presented, but many others who are in the audience already. I'll pick on this lab, for example. What is the planned strategy to assimilate data, both in situ measurements and remote sensing retrievals, into the various coupled dynamic models? And I know that different models do it differently. Some incorporate and some use them for validation or initialization. So anyone, not just Gabby, but who wants to begin this conversation, great.
I know that for CSM, we use some data sets to include sort of for like the riverine forcing. And then we use a lot of that data to validate it at the end. And I mostly focus on the like ecosystem part of the model. And so I use a lot of that observational data just to validate and be able to see if the model is consistent with what we have seen.
Joel, Mike, Blake?
Yeah, I could talk a little bit about that. So I showed the sea surface temperature data which we're using, and that's mostly, or that is for model validation. What I didn't have time to talk about was that we actually have the ability to simulate remote sensing reflectance. So actually what the water leaving gradients would be within our modeling system. And we have some limited in situ data that we collected in 2019, and the initial validations actually look quite good. We're hoping that using the radiance or remote sensing reflectance, rather than some derived products which can be relatively error prone, like DOC or chlorophyll A, can help us validate the model in the future in terms of the actual optical properties. But that's work yet to be done. Hopefully in the next few years we'll have something out actually looking at simulating these reflectances and comparing it with satellite data.
Joel, anybody else? I'm not thinking, no, just
sure. So one aspect that it's not assimilated into the model directly that Andrew Robertson working on related to the sea ice is to have a satellite emulator that you can fly through the model and pull retrievals that match the satellite record to do actual validation of the satellite retrievals for sea ice, along with what the model predicted sea ice is. And that's a framework he's developed for the validating the sea ice model. I don't know how amenable it would be to other things such as the CDOM or other aspects that if you could predict that to come through it, you'd have to come up with the metrics that you could read it off of. Also, I don't know if there's other folks here who work on the E3SM model who may have a better insight on some of that could speak up, but I don't know of direct data assimilation related to some of these ocean issues that are currently capabilities in the model.
Yeah. Any other comments? Anybody other? No, I know there are other modelers in the audience as well.
Joaquim has a question in the chat.
Joaquim has a question in the chat. Oh, I see it. Thanks. Joaquim, do you want to unmute yourself and then ask it directly, or I can read it?
Thank you, Patty. Yeah, my question is about potential for hypoxia and ocean acidification. Do any of the models include dissolved oxygen and the carbonate chemistry as well?
I'll just answer quickly. So yes, we do include dissolved oxygen currently in the model, and that is impacted by the degradation of organic matter, and as well as sediment diagenesis and the uptake of oxygen into the sediment. With response to the carbonate system, that's the future project that's being led by Maria Giorgio. It's under the NASA Carbon Cycle Science program, where we're going to build in the carbonate system for our Yukon model. And like I said, the Arctic GRO data set is key to that, because they have the ability, they give us the ability to actually load the model with the variables such as total alkalinity and pH to complete the carbonate chemistry system.
Thank you. Do you think that hypoxia could be a problem?
That's a good question. I couldn't say yes or no one way. I mean, at least in the Yukon outflow, the stratification is very strong, but there's also very strong mixing because of this really large sort of density-driven flow that's occurring and shear that's occurring across the picnic line within the plume. Maybe later in the summer when there's not so much, you know, when obviously temperatures are higher and you've already had this large deposition of organic matter into the bottom waters, that could be an issue, but I actually haven't looked at it yet. So that's something to do.
So I see that Nicole Jeffrey indicates that the DOE E3SM model includes oxygen and carbonate chemistry and has the capacity to simulate hypoxia. And I think that the CESM does as well, though at a much coarser resolution. It certainly does in other places. Don't know whether it's been tested or tried for the Arctic specifically.
Yeah, I also, I agree with you. I haven't checked in the Arctic specifically, but I know it definitely can simulate it overall.
All right, well, thank you. No, we're ready now to go to the breakout groups. And we have a set of questions, and I'm gonna share my screen just to give some, let me see, that's not what I need to do. I need to be over here and I need to share screen and go here. And so OCB and Zoom, you will be assigned randomly to a breakout group. All the groups will be addressing the same questions, and there will be probably a link that will give you access to those questions. Please introduce yourselves at the beginning of the, when you join your group. Some folks didn't discover who they were until the end of the group yesterday. So we're trying, this is as much to answer the questions as to provide an opportunity for networking among a group of interested scientists. For each group, please designate a person who takes notes so that you can report very briefly, 3 minutes at the most, when we all get together. And then please send us your notes. It doesn't matter. You can take a photo to your handwritten notes, like mine were yesterday, or type them in directly into the Google Doc. With that, one of us will remain behind in the main